So how many people have heard of DNA methylation? Heard of it? How many people are heard of DNA methylation age? All right, perfect. A little bit, yeah, familiar. So as Carl mentioned, a lot of my work is focused on social policy and economic policy, but I like to think about how those things happen and how aging happens. And so this lecture is a little more on the biological side of things. But the context of what I want to talk about today is how epidemiology can contribute to those understandings. So there's three objectives, three things I want you to hopefully understand a little better at the end of this 40 minutes or so. What is a biomarker of aging? Why would we care? What is DNA methylation age? Why would we care? And then how epidemiology and quantitative social sciences may contribute to our understanding of those. And if those aren't a little bit clear to you by the end, please ask questions. I'll cover sort of three of my papers. I'll talk about a number of papers, a number of literature, but this will kind of focus around three papers that I've published, which the first one is really a demography paper, then sort of a clinical medicine paper, and then more of a molecular biology paper. But they're all along this same theme. So there's this fountain of youth idea that people seem to be obsessed with. Maybe that's more in the US. I mean, we tend to be very obsessed with death and dying to the exclusion of sort of, quality of life. But I think even in the US, people still care about quality of life. And I think when it comes to aging and the idea of aging and biomarkers of aging, those aren't necessarily just about mortality or how long you live, but sort of the quality of life as well. So it's been this kind of obsession of the Aging Institute and of NIH for a long time to try to understand why age is so related to all of these things that we see. So this is the sort of discipline of aging research and gerontology. And it's something as an epidemiologist that for a while you just take it for granted. You stick age in your model, you ignore it. It's a really strong nuisance parameter. But the whole idea is to understand why. Why is it so important? We have the strongest risk factor for pretty much any disease. And it's about unpacking why that is to help us understand disease. So there's a number of different definitions of biomarkers of aging, but this is sort of a useful one. I'll just let you read it. The key thing here for that biomarker of aging is that it's even better than chronological age. So this is where sort of the title of my talk comes in, that accounting for chronological age, this biomarker would tell you if you're sort of aging faster or slower, this sort of category of biomarkers, than your actual calendar age. The problem here is we don't have a gold standard like we do for sort of clinical outcomes or deaths. So most of the time we're thinking about how does this map on to calendar age, and then what is the variance in terms of sort of your aging biologically as compared to your calendar age. So this is a lot to read. I mean, I don't really want you to read through all of it. You can, but just to say there's been these definitions of sort of, it's impossible, I know, you have to read. These definitions of how to come up with a biomarker of aging. And first I want to focus on some of the latter ones. It's something that's easily measurable, that doesn't kill the organism. So for example, if we're interested in sort of cognitive decline, we may love to have a biomarker taken from brain biopsy, but that's sort of off the table for this sort of realm of understanding. We're trying to look for something systemic, something from blood, something that we can easily sort of assay. It should track with time. And then I want to focus today on really definitions 2 and 3. And again, this is one of many definitions, but it's what a lot of people come back to in kind of thinking about what defines a biomarker of aging. And so that point 2 up above is that it's not a predisposition. So this isn't something that's genetic, that people are born with for how quickly they will age. So by definition, something environmental. The third point up there is that they talk about something that's reproducible in cross-species comparison, so something very universal. And I've taken this even a sort of a step back and said, if we use epidemiology and we think about this point 3, do we see the same relationship in these biomarkers of aging, or these possible biomarkers of aging, across different population groups, people living in different countries, different gender, different racial ethnic groups? And this can be one of the kind of epidemiologic contributions to this question. So not even saying species, but if we want just sort of a biomarker that we think is going to be useful, that's something sort of inherent to humans, we should see this sort of replicated in different populations. So I want to give an example of this and how this can be useful from an epidemiologic perspective. So HDL cholesterol is one of these sort of classic cardiovascular risk factors that really differs by gender. So it's universally thought, at least in clinical medicine in the US, women always have higher levels of HDL cholesterol, which is sort of protective for cardiovascular disease. And even such that sort of cardiovascular risk scores adjust differently for HDL cholesterol depending on your gender. So this is a great paper in AJE from about 20 years ago, just showing how that sort of, what people think of as this fixed sort of sex-based relationship actually differs by country. So the ratio in Canada and the US is sort of what we think of, but if you look across other countries, there's a much closer similarity between HDL cholesterol between genders. And so this isn't something, this would suggest that it's not sort of a biologically fixed or sort of a sex-linked trait, but it's dependent on context. So the first paper I'm going to talk about is sort of more of a demographic paper, and it's taking this idea to biomarkers of aging. So a bunch of 12 different biomarkers I'll talk about in a minute, and saying just a very basic descriptive demographic paper. How are they even related to age overall in the population, by gender, by education, and by place? And so I picked these sort of 12 biomarkers that are risk factors for mortality, for cardiovascular disease, that are thought to be potential biomarkers of aging. So this is just these plots. You have age on the horizontal axis, level of the biomarker on the vertical axis. And some of these sort of for HDL cholesterol and telomere length, it's sort of higher is worse. So when you look at these just related to age, they all have a relationship with age that's pretty clear. First thing you can do is just break it down by gender, and all of a sudden the relationships with age aren't as clear. And so this would be suggestive that perhaps systolic blood pressure, for example, diastolic blood pressure, while talked about in this frame, may not be the best sort of universal markers of aging. We can look at this in another way, where I separate by data sets and by country. And so Costa Rica here is in red, which I'll come back to later in the talk. But we don't even see a consistent relationship across countries or across populations in these. And so this says a lot of these things that we've been talking about as biomarkers of aging don't even seem to replicate across different populations. The two that do, or the one that really does, is telomere length, actually. That in this corner here, oh, sorry, this is a little blurry, but these are sort of across the populations and across gender, have a pretty systematic relationship with age. And these actually do an okay job of explaining the variance associated with age. It explains about 27% of the variance. For if you run statistical models on a regular basis, 27% of variance explained by a statistical model is like about the highest I've ever gotten before. So these are actually doing a pretty decent job, but they're still not explaining it in a super consistent way. So given the sort of relationship of telomere length, a lot of people have talked about this. This is getting to the sort of the second paper here, the more biomedical paper. Does it capture something unique as compared to these other risk factors for cardiovascular disease? There's sort of two things I'm interested in thinking about these other biomarkers of aging. One is it strongly associated with age in a consistent way, but does it make some unique contribution? So just looking at a very simple covariance matrix of all these risk factors, they're not very collinear with each other. They're pretty orthogonal. And if we look, these are just coefficients from a model actually predicting telomere length. So looking at the association between all these other risk factors and 95% confidence intervals. they're not that strongly associated with telomere length. So telomere length does seem to be a sort of independent sort of biomarkers compared to these other risk factors. We can look at it in a nonlinear way as well, and we get a similar sort of conclusion. But the point, and I'll talk about this technology in a minute, but the trouble with telomere length, and this could have been sort of a whole other talk here, and I'll just insert the sort of statement, is that after about 10 years of this sort of technology being available to measure telomere length and more sort of careful epidemiologic analysis, we don't really see that it has a relationship with mortality outcomes. So we had a large, there was, you get this kind of typical thing you see in the literature very early on, where there's a lot of sort of small studies. There's a big file drawer problem where sort of the null and negative studies aren't published. And then once we started doing larger studies in representative populations, we don't see much of a relationship between telomere length and mortality. So I think it's sort of a very useful, it's a useful aging biomarker, but if you're interested, or if people are interested in it as an aging biomarker that actually predicts mortality or predicts clinical disease, then it really sort of is less useful. And that's this idea about sort of technology and then assessing the utility of these biomarkers. So for the most part, technology is developed without a sense for sort of how useful it's going to be for sort of, that applies to a lot of realms, but thinking here specifically about medicine and prediction of mortality. And so, I think we want to sort of shrink that period of time between when technology is developed and then sort of the assessments of how useful it's going to be. And that's what's happening right now with DNA methylation, so getting finally into the DNA methylation topic. So first I'll kind of talk about what it is, and then talk about an example of some of the research that I've done with DNA methylation and how it's maybe a more useful biomarker for aging than these other risk factors. So first of all, what DNA methylation is, if you've heard epigenetics, it's one type of epigenetic modification of the DNA. And so all it is adding a methyl group, why it's called methylation, onto the DNA molecule, where there's a C and a G of the DNA sequence together. And so across the genome, there's around 28 million CG combinations that can either be methylated or unmethylated. So this is a very sort of genome-wide marker. So telomere length is just at the end of chromosomes. We don't need to go into the biology of that. But this is sort of a genome-wide, very broad sort of biological mechanism. And the way it functions is that it can enhance or diminish the expression of protein. So obviously everything happens in our body because DNA is transcribed and then turned into proteins. So DNA methylation acts as a modifier of those processes across the genome. An important thing to mention and know is that it's tissue specific. So unlike your genetic, your DNA sequence, which is for the most part similar across all your cells, DNA methylation is cell type specific. So if you have brain cells or blood cells, it's going to be different. In fact, the pattern of DNA methylation is one of the most important things that creates different cell types. So you see a very canonical pattern of DNA methylation across cell types, which creates cells to be the way that they are. And so if you think about it's primarily occurring in these CPG islands that are upstream of promoters. So if you have more methylation, it tends to sort of decrease the expression of genes. If you have less methylation, it tends to increase, although that's sort of the sort of metaphor for it. There's more details to why it matters. But the important thing to realize, it's really ubiquitous across the genome and really has a strong impact on gene expression and thus the proteins that exist in the body. So this is somewhat, it's been about a decade or slightly less than a decade since population-based studies have been able to assess DNA methylation. So it's pretty new. What happened is, there was sort of sequencing that was done originally, but now this has been sort of rapidly progressed based on a chip that's available from Illumina. The newest chip allows 850,000, so just less than 1 million CPG sites to be assayed. And then you can look at how that's related to either environmental exposures or how those DNA methylation patterns are related to clinical health outcomes. So what do we know so far about how, what in the environment, which is my primary interest, affects DNA methylation? So first of all, probably the strongest factor affecting patterns of DNA methylation in the environment is smoking or exposure to secondhand smoke. There's also been a lot of work, this is a great review to look at in terms of environmental influences on environmental contaminants. So arsenic and lead are two of the sort of first factors that have been looked at and shown a big influence on changes in DNA methylation. And then in turn, the question of how DNA methylation is related to clinical outcomes. So it was first most characterized in cancer. So cancer cells have been shown to have really different patterns of DNA methylation, which causes sort of increased transcription that's a hallmark of cancer. But it's also been shown now to be related to cardiovascular disease, pretty much any other sort of clinical sort of outcome that you could measure. So 850,000 sites, a lot of people have analyzed this sort of in a site-specific way, much like you would with a genome-wide association study. But around 5 or 6 years ago now, there was a very interesting and influential paper by Steve Horvath where he developed something that was called a DNA methylation clock. So a few people have, if you have questions about this, I know a lot of people weren't familiar with this before, please interrupt me, but we can also come back to it. So a few people had done clocks like this before, and the idea here is coming back to directly to this biomarkers of aging. And that is, can you take all of these CPG sites across the genome, do a prediction model where your outcome, your dependent variable, is calendar age, and come up with sort of a metric or a score on how these CPGs would predict your actual calendar age as a way of creating, based on calendar age itself in a data-driven way, a sort of clock of aging. Oh, I should also say an important thing about DNA methylation to know is it's not like these sites are sort of off or on within a particular person. So if you take sort of a population of cells, you'll get a score between sort of 0% methylated at a particular site and 100% methylated at a particular site. So it's this sort of percentile of methylation at one of these particular sites. So he did these prediction models across a lot of different tissues and a whole bunch of different data sets, and did a really nice bioanalytic approach of this, where he looked at some in-sample, reserved sort of an out-of-sample to sort of train his methylation prediction, used a whole bunch of different data sets from different tissues, and he found a correlation based on his clock, an R-squared of about 0.95. All right? So I'm not talking about 0.0095, 0.95 with this DNA methylation clock. So the background on biomarkers of aging, as I talked about at the beginning, since 20 or 30 years, is that people had continually been coming up with these sort of biomarkers of aging, which were really strongly associated with calendar age, and then they were kind of found to be not that useful or not that correlated or not that associated with health outcomes. So we've had sort of, in the sort of aging literature, sort of 20 or 30 years of sort of these failures. So all of a sudden this guy who's a mathematician comes along, so he's at UCLA in Southern California, and produces this sort of scale that has a 0.94 on the outer sample, 0.97 on in sample, of the variance of age explained, and of course nobody believes him. He's also sole author on this publication. So no one believes him. He gets it rejected from journal after journal. There's actually a nice paper, I think in Nature, there's a little editorial about his process in Nature, including sort of all these personal things going on, this paper getting rejected and rejected when it seems like an amazing paper. And finally, Genome Biology accepted it. To his credit, these are all publicly available databases. People have been able to replicate this and show that this is like a legitimate clock. And now people, and that this is legitimate prediction, and now people have done this over and over again, different data sets. Now I don't think there's any question that what he's found and what he's created is real. It's not any data artifact of what he's doing. So now the question is, and so there's a few questions, I think 2 broad categories of questions going forward with this. How is this clock, what influences this clock? You can kind of think about, if you think about it in a mediation framework in a way, this clock is in the middle. What in the environment and genetically influences this clock, and then what clinically does this sort of clock influence? So in terms of what this clock is associated with, I should say there's been very little longitudinal analyses. Obviously, that's something that needs to come, to get a little clear inference here. But in terms of what it's associated with in terms of clinical outcomes, it's associated with a lot of different factors. And the way people analyze this is by looking at, because it's so closely associated with age, it's sort of tricky to analyze and disentangle it from age. Like, you probably aren't even going to be doing an adequate job just using a linear and a quadratic term. And so what people do is look at the residuals from a regression model of this clock on age. And so this is called sort of, they use this term sort of accelerated aging. So how different is someone from that relationship? So the sort of error terms in a way. So this has been shown to be associated with a lot of things, even from blood. So taking blood, because it's the most accessible tissue, predicts all-cause mortality. Also, I think the interesting thing is that it's also associated with frailty measures and cognitive fitness and cognitive decline. So this marker from blood of age is associated with a lot of different outcomes. I should say that I think something I didn't mention, it has 353 CPGs. So there's around 353 sites in this model out of around 850,000. And there's nothing sort of special about that. He picked this sort of best fitting model. You can include more sites, it'll do slightly better. You can include less. There's another DNA methylation clock based on only 3 sites, and it explains around 90% of the variance. So there's nothing kind of magical about this 353 sites that go into it, but it does seem to be a useful, a pretty good model. So I'll say, so there's 2 things that we're going to look at in this analysis that I'll talk about. One is this epigenetic clock, but another is a measure of variance that happens over time with age. So the clock is about the exact percentage of these CPG sites that change over time, that are very, very closely associated with age. But there's another notion for aging called epigenetic drift, which is just the variability in your population between the percent methylation of these sites. So what you can imagine is that at younger ages, say there's a percent methylation of a particular site or a group of sites that's, say, 5%. And that's kind of optimal for health and functioning. What happens over time, and maybe sort of the standard deviation of that is like 2 or 3%. What happens over time is that variance among people sort of expands. So instead of most people in your population being methylated around 5%, that starts to spread out 5, 10, 15%. So this is called epigenetic drift, and it's sort of this dysregulation that happens over time. So we've been using this methylation. So one of the ideas with biomarkers, so this comes to the question of why would we be interested in this? Comes to sort of characterizing populations, characterizing aging. So one is using it as sort of a clinical, sort of a proxy endpoint. So instead of waiting for people to die, using this as an endpoint to look at sort of economic policies, social policies, the kind of things I'm interested in. But it's also sort of establishing like a why people in a particular group with a particular exposures may live longer. So I'm going to talk about these studies I've been doing for the last 10 years in Costa Rica and how we've sort of brought DNA methylation age and DNA methylation data into understanding that relationship. So Costa Rica, the reason I've studied Costa Rica is because it has a really high life expectancy, and it has about 1/5 the GDP of the United States, but actually has a longer old age life expectancy than the US. I mean, a lot of countries have a higher life expectancy than the US. We're around 30th in the world, but Costa Rica is one of those countries that, based on a much lower GDP, has achieved even higher life expectancy. And then within Costa Rica, there's a particular region, the Nicoya region, where people even live a longer time. And so journalists actually first found this working with some demographers and talked about this as one of these sort of high longevity regions. But people have talked, again, coming back to the fountain of youth, people for many, many years have talked about these high longevity regions, and then they've very often been shown to be due to errors in birthdays or sort of age exaggeration or a lot of different problems. So, yeah. So people tend to be very skeptical, and so there was one in Peru, Vilcabamba, that people found out really wasn't real. And really, is there even anything? Is this just some sort of statistical artifacts because people sort of look in one area? So we want to sort of characterize this area of Nicoya, see if it actually people do live there a longer time, and then see if there's sort of molecular, even epigenetic pathways of why they live there, why they live a long time. So this is this other paper from the Vienna Yearbook with Luis and Will Dow, who've been working, who started the Krellis cohort in Costa Rica, and this is all publicly available data, who I've been working with for about 10 years. And this is really work led by Luis to do these analyses. But we did a stat scan analysis of deaths in Costa Rica. And what we actually showed is that it was true. So even just taking an agnostic, data-driven approach, we identified this high longevity cluster in Nicoya, consistent with what other people had identified, with sort of a hazard mortality risk ratio of around 0.8, which is actually quite substantial. Just to compare Nicoya, yeah, you may be able to see that. How long people live in this Nicoya region as compared to other of these sort of high longevity areas around the world. There's around 5,000 people 5% of the population at age 60 live to become centenarians, and that compares to the most in sort of Okinawa and these other sort of blue zones, around 2% in Okinawa, 0.7% in Sardinia. So really an extremely high number of people surviving a long time in this particular region of Costa Rica. And it seems to be even more true for men than women. One sort of, again, taking a demographic approach here is that it doesn't seem to be the case. And again, this is totally descriptive. Obviously, people who migrate out of Nicoya, migrate into Nicoya are not a random sample of the population. But if we just look descriptively at, was there a sort of longevity and life expectancy advantage for people who moved to Nicoya or people who moved out of Nicoya? There was sort of no advantage. This was something people needed to live there for their whole life. We looked at a number of sort of normal risk factors for disease, and they really do not explain why people in this area could live longer. And so, on some sort of risk factors, people in Nicoya are worse, and in some they're better, and it all seems to just kind of balance out, including smoking, lipid levels, all of that. So it can't really be explained. And this is another paper by Luis and Will going over this in more detail. Can't really be explained by sort of known risk factors. So this is why we're interested in, can DNA methylation sort of help us first, better understand, is there something different about this population and what might it be related to help us kind of think about, really as a wedge to kind of think about what the environmental causes may be there. And so we have this paper that you can look up the details on. The most interesting thing we found is sort of the different sort of immune function and cell types related to people in Nicoya. So they had more naive CD8 T cells. So this is shown, Nicoya is shown in blue here, and fewer of the, the Nicoyans are in blue. They had more naive cells and fewer memory cells. So less of an exposure to infection that we captured. So this is capturing it in later life, but this is sort of a cumulative impact of sort of what their lifetime sort of exposures were. And this was even more true for people as they tended to age. So again, like what I talked about, this is, methylation data is useful in a lot of ways. Certainly the clock, which I'll talk about in a minute, and what it looked like here. But also for identifying cell types from blood. Typically you would have to do a cell sorting, which is super expensive. You need a lot of cells, but you can get specific cell types because every cell type has such a sort of canonical methylation pattern. So when we looked at these different clocks, these aging clocks that we're very excited about, we didn't see any difference between Nicoya and the rest of Costa Rica. And I should say this is in a very small sample size of about 100, and we now have a larger sample size that we're looking at it in, but results look about the same. So we don't see any difference in that DNA methylation aging clock. One of the interesting things we did see, shown on the bottom figure here, is just the difference overall in the clock between Costa Rica and the rest of, and on the samples that have been trained on, which are primarily US and European samples. The Costa Ricans on average are about 7 years younger in terms of their DNA methylation age overall. So we did see a really big difference in the Costa Rican population versus the other population. But where we did see a real difference was in this variability. And so if we look, this is a measurement sort of the interquartile range and variability across all of these CPG sites, with Nicoya shown in blue here and the rest of Costa Rica shown in black, with age on the horizontal axis. So over time, you see variability in both populations, but you see sort of a dysregulation of DNA methylation in the overall Costa Rica population as compared to Nicoya. So we saw, we also looked site specifically, and we saw a lot of differential methylated regions. One of the things that you can do here is, instead of just looking at one site, you can look at a lot of sites upstream of a gene. So it's unlikely to be sort of a useful marker if there's only one site that was methylated. So you can look at a few different CPG sites and see if they're all consistently more or less methylated. But again, this was based on a very small sample, so we don't want to make a lot of inference yet about these sort of specific genes that are influenced. I think the variability in the score is something that you can do in smaller samples. Another nice thing, just on a sort of, if you're interested in this, you can do, and this is kind of from an epidemiologic perspective, that we're really interested in how these things can be replicated. So the chips themselves are pretty expensive. So the reason there's these small sample sizes is they're still around $250 per sample. But what you can do is you can go in and assay specific sites from an out-of-sample population for around $10 and see if what you found in your sort of main sample replicate in another sample. So I'll sort of conclude here with sort of two kind of overview slides and then see if there's questions or things you're interested in discussing. I think it's always difficult to make, well, not I think, it is always difficult to make comparisons or any inferences between sort of two populations, you know, Costa Rica and somewhere else, or Nicoya and somewhere else. So we've gotten samples from some of these other high longevity populations from Macquarie and Sardinia. And we're in the process now of looking, is there sort of a similar DNA signature or DNA methylation pattern or aging pattern associated with longevity in these other sort of high longevity regions? And that gets at the overall idea, is there some unique thing that allows people to live longer, live healthier in these regions? Or are each of these regions achieving higher longevity, higher life expectancy in their own sort of unique way? I think obviously having a larger sample size is really important, which we have 500 now, and we'll have another 500, so we'll be up to 1,000 by the end of the year, and that's, you know, really important for doing any of these sort of analyses, obviously. Genotype data is something that's important as well, so DNA methylation is also very influenced by genotype, so being able to disentangle you know, environmental from genetic influences. You know, again, this was all very descriptive work, but it's hard to sort of think about anything other than genetics that would kind of be a confounder to think about between sort of Nicoya and the rest of Costa Rica. So primarily it would be some sort of population stratification that we're worried about. You know, people have done work on that so far and haven't found any sort of genetic differences, but it's really been kind of based on pretty old data, so we'll be able to assess that more accurately. Then the other thing is trying to bring in more historical exposure data. I mean, I think some of the things with telomere length or any biomarker, the great thing about those is that they're really capturing the long history of what the human has been exposed to. So we're thinking about aging here, but this is sort of representative of what people have been exposed to over their whole life course. And if we want to understand the environmental impacts on the changes in DNA methylation, it's important to have sort of exposures from earlier periods of time. And so we're really looking to find more sort of quasi-experimental exposures. So when there was sort of a lack of food in different parts of Costa Rica over time, when diet changes, when other things were introduced, weather patterns at particular periods of time, there would be sort of have a quasi-random or as random as possible spatial and temporal variation to sort of get at more causal questions. And then I think, from an epidemiologic perspective, I mean, I mentioned some of these, but the replication of multiple samples, the idea of finding quasi-experimental exposures, and then, again, coming back to some of my earlier points, like so much of this work is assumed to be fixed within a particular cohort and a particular culture, and the more we can get outside of sort of the Western, Western European and United States samples, there's really a lot more I think we can learn about these relationships. So a final slide here before I conclude. I think that one of the conclusions I've come to is that there's a bit of a tension between the sort of criteria 2 and criteria 3 for biomarkers, and that is that something that is not variable across species is, to me, more likely to probably not be environmentally impacted. So I think there's some questions about how we need to revise our definition of biomarkers of aging, and that this sort of idea of cross-species comparisons may be something to sort of drop from the definition, or at least have a sort of biomarkers of aging in humans as a separate type of road that we go down. And then instead of having this idea of cross-species comparisons, sort of cross-country, cross-national, cross-demographic group comparisons to really validate from a demographic and epidemiologic perspective the way we use these markers. So thanks very much for listening. Hopefully that wasn't too much molecular biology for you, but I look forward to your questions and comments. Thanks.